See, one thing I am telling you repeatedly, reading one time will not solve any problem. We have to read at least 20 times. I used to read, write even answers on 20 times on a paper before I used to go to the examination for anything. It's not that easy, it is not that easy to remember, and your tenure is only 8 months, not 1 year also, to be very frank and honest. And time will run out, I am repeatedly telling you. In no time you will appear in the first internal assessment. So when you will read, and before failing any students, only simple questions are asked. See, if you cannot answer, if you do not read, then there is no purpose of teaching you. My purpose is not to allow you to remember everything. My job is to stimulate you to read and to think something else. Everything is written in the book. What is the purpose of attending a class? My job is to create some amount of interest and to stimulate you, so that you read, you ask some questions which you do not, for which I am sorry. I would, on the very first day I told you very clearly that I would really love if I cannot answer to that question, then I will have to read. I am not a Pandit. I am in the learning phase and I want to learn from you. You are the best teachers. Yes, all the students are the best teachers to the, you know. If you do not ask any question, then where from I will get the input? What will be my food from the class? So, yes. So, I will discuss about today protein digestion and absorption. Have you read about chemistry of protein? How many of you have read chemistry of protein? Okay, 1, 2, yes, 3, 4. So, you have not read chemistry. So, if you do not know the chemistry part, then again it will be difficult. See, in the chemistry part, what has been told? That proteins are basically polymers of amino acids linked together by peptide bonds, and proteins are having primary structure, secondary structure, tertiary structure, and in some cases quaternary structures. So, that is how a functional protein is formed. Now, in that process, all the linear peptides are really folded into a globular form or a fibrous form, and the peptide bonds are almost, most of the peptide bonds, almost nearly more than 90% of the peptide bonds are inside that protein molecule. Now, when something is to be digested, then that protein part, that primary structure is to be opened. If the primary protein remains in the quaternary structure or tertiary structure, in which it remains normally, becomes functional, then you have to undo the entire process to get the primary structure. How can we do that? See, normally during our infancy, we take protein in native form, like from mother's milk. We do not cook it because our system is acclimatized to that. But as we grow older, we take protein which is cooked, which is exposed to heat, which is exposed to boiling. During that process, the protein undergoes denaturation. And during this denaturation, that unfolding of that tertiary structure into primary structure, that all these structures are lost, the primary structure is opened. And that is the reason protein digestion becomes easier if you cook it properly. Uncooked protein, it is very difficult to digest. Partially cooked protein like meat, fish, you really cannot digest, because that structure, some of the protein structure will remain in that form. So, the basic purpose is to denature the protein and expose the peptide bonds which is within the protein. Now, what happens basically, that you will see that protein structure I told you, that it is having multiple amino acids. This structure is now opened. Now, as you know, a protein is a very large structure. So, if it is a monomeric protein, so one single polypeptide chain will be there and that will be folded. Now, if it is a multimeric protein, then subunits will be formed. Now, this structure is formed. So, these are peptide bonds. So, either how we will get this thing digested, how we will get it, either basic purpose is not digestion, we have to absorb. Whatever we have food we have taken, we have to get that into our body. So, if I digest it and if I do not absorb it, then the protein, there is no purpose of digestion. The purpose of digestion is to absorb the material, what has been digested. So, basically we have taken the protein, so our basic purpose is to get each amino acids into the system. So, how you can do that? Can you break by any machine into individual amino acids? Is there any machine? You can grind. You can grind the protein to a lower fragment structure, no doubt about it. You can grind it. You take a protein and grind it. But you will not get the intact amino acids. If you make whatever powerful machine is that molecule might be destroyed, but that intact molecule, the intact protein, amino acids will not be there. So, that is accomplished by enzymes. So, what enzyme does basically, they cut different sites, and this large polypeptide molecule is broken into several small species. That may be a polypeptide, that may be an oligopeptide, and still it is a tetra, tri or dipeptide, even amino acids. So, proteins are normally formed not initially in the polypeptide stage, there may be first stage is proteose, partially digested protein. From proteose you will get the peptone also. Then from there you will get that thing. Basic purpose at the end we have to get the each amino acids from the protein. So, once the digestion is complete, then finally we will get this one. Otherwise, during the intestinal tract, partial digestion will occur in the stomach, as we will see in the picture. I will diagrammatically show it to you. So, we take the food in the to the mouth, there is no basically digestion of protein in the mouth. Protein digestion starts from the stomach. Now, in the stomach, as you can understand that after we take the food, it remains about 3 to 4 hours, even 5 to 6 hours maximum. After 4 hours, if the food remains in your stomach, and if you belch it out, remember that food is not suitable for you. If you take a normal amount of food, ideally that much amount of food must get out from the basically stomach into the duodenum, or maybe a small amount, if you take adequate amount of water and that normal food. But you have taken excess food many times, and you have taken unusual foods also, that you have realized that still I am not hungry, it is not empty. Anyway, that is altogether different issue. So, after the food is emptied from the stomach into the duodenum, some digestion also takes place in the duodenum, then jejunum, then ileum. Now, from the ileum basically absorption takes place. So, that I will tell you in detail. So, this part you understand, only some part I will tell you to write down. So, your basic syllabus right now contains describe the digestion and absorption of dietary proteins. So, this is your topic basically. So, digestion can occur the mouth, stomach, intestine, 3 parts as you know, duodenum, jejunum and ileum. Large intestine digestion usually does not take place. There are bacteria which are not of our concern. So, these are the sequential exposure. You can see, can you read? You cannot perhaps. So, in the mouth first the food is chewed, that means that large molecule is broken down into smaller fragments. That protein molecule or food molecule, whatever you take, either in the form of protein, fat, carbohydrate, does not matter. We are breaking it down, so that we are exposing many sites. Now, that is grinded also, broken down, grinded, and then it is passed through the esophagus into the stomach. In the stomach, basically there is a H cell. What is the function of the H cell? Function of the H cell, I will tell you that first of all it creates acidic medium. We do not have an acidic medium in our mouth, otherwise I mean strongly acidic, it is acidic definitely, slightly 6.0 to 7.03, this is a pH, but stomach is acidic. So, in that acidic medium, medium is due to hydrochloric acid. Now, that hydrochloric acid basically causes denaturation of the protein, and it also sterilizes. It's actually a disinfectant. So, normally with the food we may take some amount of bacteria, and we are likely to get infected from that bacteria. Many times what happens, you do not wash properly or the foods are not cooked properly, some bacterial contamination is there. So, stomach H cell basically destroys that thing, and also it activates pepsinogen to pepsin, that I will discuss. So, after that digestion by the pepsin is complete, then it will be thrown into the duodenum, where the enzymes are there. I will discuss only restrict my talk only on protein discussion, not others. Then it will pass through intestine and then absorption will take place. Finally, have you been told about portal vein anatomy in the anatomy class? Portal vein, intestine has not been told in the anatomy. Then it is difficult, because see, intestine, everything whatever is we are taking, the drainage area is basically, draining vein is portal vein. So, whatever food we are taking, be it carbohydrate, fat, protein, finally once the absorption takes place, it gets absorbed through the portal vein, and portal vein enters into the liver. So, it directly delivers into that liver. So, it is just like the storehouse of everything. Then liver decides what is to be done with that, whether we will reserve, synthesize in a storage form, or we transport it. So, basically from that portal vein, that means that food is absorbed by the intestine, and intestine it will deliver into the bloodstream. Basically that amino acid has to reach several parts of the body and each and every cell, because the cells will synthesize protein, and the building blocks of protein is amino acid. So, in summary, what I am going to discuss, I have told, in a living being, breaking the quaternary structure, tertiary and secondary structure, I have told the first step, and then sequential exposure of polypeptide chain to protein digestive enzymes like proteolytic. All the proteolytic enzymes are hydrolytic, because they break the peptide bond by hydrolysis. So, all the enzymes are essentially hydrolysis. So, cleaving of protein into smaller units, as I told you in this class, into peptone in the gastric juice, to proteose. Usually, the dipeptide, tripeptides are not formed in the stomach. Polypeptides, then oligopeptides. In pancreatic juice, finally to tripeptide, dipeptide, and individual amino acids. Saccus entericus means intestinal fluid. If you do not know the name of intestinal juice, then write down Saccus entericus. Now, absorption takes place to the small intestine border, that perhaps you have read during a class 12 class. So, this you note down, just have a, just look at, you need not note down because we are not talking about fluid homeostasis, but what happens basically here, you see entry normally we drink water normally because we are in an extreme environment. In the summer we take almost more than nearly 4 liters of water, in the winter sometime around 2.5, 2 to 2.5. So, on an average I am telling you, this is from foreign book. So, they take maybe a less amount of water because of the temperature is quite low. So, saliva water content is 1200 ml, saliva water we take with the food, and saliva 1500 ml, stomach secretes 2000 ml, pancreas 1500, bile 500, and in the center per day, that means this much amount of fluid is entering into the system, into the digestive tract, because we require an aqueous medium for digestion to occur. Otherwise, if you do not take water, you cannot digest food. You have noted it. You take less amount of food, food will not pass from the stomach, because that water basically mixes. Can you see, with saliva per day 24 hours we are secreting almost 1500 ml, and stomach itself 2 liters. So, if you do not take water, what the stomach will do? Stomach will draw water from the blood, and in the plasma will be concentrated, and you will feel thirsty after some time. If you do not take adequate amount of water with your food, after 2 to 3 hours you will feel thirsty, because stomach will drag the water. Stomach will drag the water from the plasma, and plasma will be concentrated, and that will stimulate your thirst mechanism to operate. That is the basic reason why we feel thirsty. When the osmolality of the plasma will be high, there is sensor in our hypothalamus that allows us to drink water. Similarly, exit portal vein, I am telling you, this huge amount out of 800, 8,000, 6,700 is absorbed, and resorption through colonic resorption is occurs about 1049 ml through stool we pass, because we do not pass absolutely hard stool. Stool is soft, semi-solid. Why semi-solid? Because some amount of water is there along with the undigested foods. So, that amounts 800. So, that is the basically at the end we are getting 100 ml. Of course, I have not included some water we are exhaling through our lungs during respiration, that you have noted. You exhale in front of a mirror, you will see that water vapor is coming out. So, just note down this one, pH of different juices for your understanding and remembering. So, saliva is usually acidic, it has got no connection with our protein digestion. So, gastric juice, remember, it is strongly acidic, its pH is 0.9 to 1.5. In biological system, there is always some amount of reference ranges, because you can see in your class all persons are not unique. There will be some variations in individuals. Now, that normal variation of anything, any parameter, not only pH, let us say blood glucose or say blood urea, creatinine, whatever it is, there is just some reference range. We cannot have an absolute value, just like 1 rupee, 2 rupee or 50 paise. So, gastric juice is strongly acidic and pancreatic juice is strongly alkaline. You can see the note on the pH, neutral pH is 7. And Saccas entericus, intestinal juice, is 6.3 to 9, it is average 8.3, it is not that high. But depending on the type of food, whatever you are taking and what is the acidity, that pH also changes. Just only note down this point, and I will go hurriedly next to the next one. See, zymogens. Have you been told about zymogens during enzyme class? Definitely it has been told. Zymogen, do you remember? Any one of you remember? See, normally these type of enzymes are not secreted as such in that active form. Just like if you remember P-pro-insulin, insulin is not secreted directly in the insulin form, there is a P-pro form. So, pre is cut, then pro is cut, then finally we get that insulin. Similarly, enzymes are also formed in an inactive storage form, that is stored in the zymogen granules, and this is secreted on stimulus by exocytosis. You know what is exocytosis? Can anybody explain what is exocytosis? Otherwise, I will be speaking only one way. And if you do, what is exocytosis? Can anybody explain? Try to explain what is exocytosis. These are the simplest questions these are asked. You just tell something you try to tell. See, I do not want to waste time because, see, exocytosis, endocytosis, have you note down, you just read it. Otherwise, you ask me in the next class, I will explain it to you. Then are activated by proteolysis, and that is the reason why you are not, see, if you do not know the basic things, you cannot understand. Activated by proteolysis means that particular protein molecule which is larger, some part has to be broken down to make it active, because the active site of the enzyme are usually masked by another protein. So, that part has to be, or a small polypeptide chain, that has to be clipped off, so that the active site of the enzyme is exposed, and that is what is activation. Note down this one. First, go on first, this is basic part. So, from zymogens to active form, this I will be telling you repeatedly, but you note down this one. Pepsinogen. Pepsinogen is the zymogen form, active form is pepsin, and it is done by initially by H cell, initial part. And once this part is done, then there is a autocatalysis. Remember, pepsinogen is initially activated by hydrochloric acid, and then there is a autocatalysis. As I told you in the beginning, the role of H cell is basically denaturation, and it is also an antiseptic. As I told you, with the food, whatever bacteria we take, or bacteria, virus, or anything we take which is undesirable, that is destroyed. So, one thing I am going to explain to you, this pepsinogen is basically zymogen, its molecular weight is 44,000, okay. Now, that some part is, the active site is masked by a polypeptide chain, that is cut, and you can see the molecular weight is 32,700, it has been decreased. Similarly, trypsinogen is activated by the enzyme enterokinase, or it is called enteropeptidase. Write down, enterokinase and enteropeptidase are same thing. It is present not only in duodenal cell, but in intestinal cells also, epithelial cells. So, what this enterokinase does, it removes one hexapeptide. That hexapeptide masks the active site of trypsinogen. So, once that is removed, then the active site will be exposed and trypsin will be active. Now, once this trypsin is formed, trypsin actually catalyzes trypsinogen, chymotrypsinogen to chymotrypsin, pro-carboxypeptidase to pro-aminopeptidase to carboxypeptidase and aminopeptidase, and pro-elastase to elastase. The trypsin alone does it. So, activation of trypsin is very much important. Activation of trypsinogen is done by enterokinase enzyme. Note down, I will schematically show this thing in a short while. Please be fast, because otherwise it is 9:30. Now, proteases are present in the intestinal mucosa. These are enterokinases or aminopeptidases. At this today, this is a very simple thing. Today, after I mean, completing your class, you just finish it so that you remember today, you read it from the book. Okay. See, this is intestinal brass border, be it jejunum, there is enterokinase enzyme is there. Now, trypsinogen is there, chymotrypsin is there, procarboxypeptidase is there. So, now this is being acted. So, you can see trypsinogen is converted into trypsin. Now, this trypsin basically converts chymotrypsin to trypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase. Do you get my point? What I said in the first class? In the first point, whatever I said, you just note down and try to, check this thing in the image. Whatever you have written. This one, most important slide, because a short question or MCQ will be given. Which site is cleaved by which enzymes? And this information has been taken from Harper books. There were, several confusing statements are there in different books I have seen. So, this is the 1232 edition of Harper, this page number perhaps 591. This I have taken it from that side. And I have also edited a little bit to make you understand. So, remember one thing, pepsin first. One thing I forgot to tell you. Endopeptidases. What is endopeptidases? Means the peptidases, basically all the enzymes, proteolytic enzymes, peptidases. So, peptidases basically cause breakdown of peptide bonds. When that inside bond or internal bonds of the polypeptide chain is cleaved, that is done by the endopeptidase. And at one end, that is N terminal or at C terminal, at one point of time you will get a dipeptide. So, that part is cleaved by aminopeptidase, amino terminal end. Similarly, this part is called by carboxypeptidase. This is exopeptidase. So, endopeptidase cleaves the internal bond in the peptide bond, and exopeptidase cleaves the extreme outside, that is one at the carboxyl terminal and one is amino terminal. So, pepsin, you just note down very clearly. So, cleaves the peptide bonds of the carboxyl group of aromatic and branch chain. In addition, there is a methionine also. To remember, simply I have put this thing because here you see aromatic and branch chain. Here only aromatic. Trypsin, it is basic. And elastase, small neutral. Remember this thing. First one you remember aromatic branch chain, second one chymotrypsin, that aromatic branch chain part is out. Trypsin, remember, it is basic, and elastase, small neutral amino acids. Endopeptidase I have told you, exopeptidase I have also covered. So, at the end what happens? Basically, you will get a dipeptide or tripeptide as I told you. So, these are the names of the enzymes, you just write down. So, tripeptide is this one, tripeptidase will cleave this part, this peptide bond. Dipeptidase, it will cleave this one, aminopeptidase, that means N terminal left side. Always amino terminal is written on the left side, carboxyl terminal on the right side. So, there are two types of aminopeptidase, one is leucine aminopeptidase, that means cleaves of only what is remaining in the leucine and proline. Similarly, carboxypeptidase is on this side at the extreme right hand. There are two carboxypeptidases, one is A for aromatic and amino aliphatic, remember. A for aromatic and aliphatic, but it will not act if the C-terminal residue is arginine, proline, and lysine. So, that is specificity. Similarly, carboxypeptide is B for basic, remember. A for aliphatic and aromatic, B for basic, and it will act, remember, it will not act, it will act if the C-terminal is proline. Here it will not act if the C-terminal is proline. Here it will act if the C-terminal is proline. So, that is the specificity. So, mouth, as I told you, there is no digestion in the oral cavity, no proteolytic enzyme. Protein digestion starts from the stomach. It acts as a hopper. This particular word I have taken from your medicine book, because you will be reading medicine at the end in a short while. So, its basic purpose is to retain and grind food and then actively propelling, because have you been told about stomach, anatomy stomach? So, then it is difficult, we have to basically correlate with, if you know the anatomy, then it will be easier for you to understand. And that is why I have taken also anatomical picture also. So, remember one thing, gastric secretion, the key stimulus is 3 hormones. One is gastrin, histamine and acetylcholine, write down. This histamine is having a clinically important point, because antihistaminic, H2 receptor antihistaminic drugs are given in peptic ulcer for indigestion, and many of you have taken ranitidine, famotidine, you have taken, you have been prescribed perhaps in your lifetime, you have taken. Hydrogen and chloride ions secreted from the apical membrane of the gastric parietal cells, and it is through proton pump, that is hydrogen potassium triphosphate proton pump. I am sure all of you have taken omeprazole for some amount of acidity, without knowing what it is, what it does. It basically blocks proton pump. So, one part stomach also secretes, that is glycoprotein intrinsic factor, and that helps in the formation of absorption of vitamin B12. So, this is the structure of the stomach, remember. So, I told you it is acting as a hopper, it is acting as a storage site. This is a large structure, food is stored here. Now, once the food is partially digested or formed, then it is propelled. That hopper means, expulsion with some amount of force. So, there is a sphincter here. Since this the anatomy has not been told, so normally that sphincter remains closed. It does not allow the food to enter into the duodenum. Now, once the food is almost ready to be propelled into the duodenum, then only the valves of this pyloric sphincter opens, and it allows that part, not the whole amount. It gets closed, then another part. So, gastric juice, pH strongly acidic, I told you, it is a mixture of secretion of stomach. There are 3 things are there: HCl, pepsin and mucus. Where from HCl is secreted? It is from parietal cell. Write down. Parietal L means HCl. Parietal L means HCl. Remember. And chief cells, pepsinogen. Columnar cell, mucus. I will tell you in the histology in a short while. Now, function of the mucus is very important. It protects the epithelium. If that protective epithelium is destroyed by some reason or other, ulcer is going to happen. And that can happen if we take some foods which will destroy the mucus, or some drugs which are taking which will destroy that mucosal barrier, then it will cause acid peptic digestion. So, it protects the gastric mucosa from acid peptic digestion, and gastric acid, peptic ulcer, gastric ulcer, duodenal ulcer results from overactivity of acid peptic digestion. So, breakage of the barrier, then it will cause gastric ulcer or duodenal ulcer or peptic ulcer. Peptic ulcer is a general term where acid peptic digestion is involved. So, as you know all that for any enzyme to act, optimal pH is required. So, pepsin will act in the acidic pH. Similarly, chymotrypsin, trypsin will act in the alkaline pH. So, they will not act in the acidic pH. So this is the histology. You can see the three things. Simple columnar epithelium is remaining on the top. It forms a mucus. It forms mucus here, and parietal cell, as I told you, it is L means HCl. Remember, L means HCl, so it secretes HCl, and chip cell produces trypsinogen. So I am amplifying this thing. There are three things I told you: surface epithelium, parietal cell is this one, chip cell is here, and enteroendocrine cell. You know, all of you know that about the endocrine glands, that pituitary, thyroid, adrenal, and largest organ, endocrine organ. If I ask you, they will say that it is thyroid. But gut is the largest. Human gut is the largest endocrine organ. Before 2000, it was not known, honestly speaking, that gut can produce so much hormones. So, gut produces nearly more than 20 hormones, and these are very important hormones. Gut is the largest endocrine organ, if you say, as a whole, collectively. So, these gut hormones basically regulate the secretion of intestinal GI tract. So, this is the opening of the gastric glands. Once you read this anatomy histology, you will understand it better. So, opening this pepsinogen, chip cells are producing pepsinogen, and H cell is produced by the parietal cell. Now, this H cell is now activating. As I told you, that pepsinogen is activated by H cell, and then this H cell autocatalysis is there. So, that is schematically being shown here. Okay, so protein is basically digested into peptide fragments. So, acid secretion is a very complicated process. Whatever you are thinking, it is not that. Most of the gastric secretions or secretion of the stomach are controlled by the brain. See, if we smell, if we hear some food which we like, there will be some secretion. Similarly, anxiety, over-anxious patient, they secrete some amount of acid, and usually when the patient suffers from gastric ulcer or duodenal ulcer, in clinical years, you try to understand the psychology of the individual. I am telling you, the most of the common diseases, whatever you will encounter in your clinical practice, spend a little time to understand the psychology of the patient. And believe me, all the patients, almost as a rule, almost all the patients are suffering from some kind of stress or strain. And that is manifested in different ways. And one of the manifestation again, expression of that tension, stress through the GI tract, either in the form of diarrhea, what we have noted, perhaps before examination, you go to the toilet. Why? Because there is some amount of action is there, you are having tension, that is causing anxiety, that is causing intestinal movement, rapid intestinal movement. And many of the women might have some menstrual disorder also due to anxiety. Similarly, the entire body is working under the influence of brain. So, control of acid secretion, you will see it is not possible to cover in this brief time. So, just I am schematically showing you how complicated it is. That simply we are taking food and this HCl is produced. It is not that. There are so many hormones are acting on these cells. I have told you only one. But critical interplay of many things are required. So, an integrated system, integrated biochemical system will result in normal acid secretion or normal functioning. And the more you know, the more you will realize that emptiness. Earlier when we were students, MBBS students, this part was not known at all. So, therefore, we are very much happy with that information that stomach secretes HCl and pepsin, that is the end of it. It is not that. So I told you this part, again I remember that chart. Renin, there is a confusion. Renin is formed ideally in our student life, even in your textbooks I have seen, renin is told that it is present in the infant, but in humans, being renin is not present. It is present in the cud chewing animals of in the fourth stomach, like cows. It causes insoluble casein formation from the milk protein. So it calls basically calcium paracaseinate. So that helps in the digestion because that is an insoluble form that remains within the stomach for a long time. So if you see the endoscope, normal endoscopic view of the stomach, if you put an endoscope, you will see a structure like this. Now the whole purpose of telling this gastric acid secretion is basically, you will clinically see patients are coming with some indigestion, some form of stomach pain, abdominal pain, indigestion, and if you put an endoscope, you will see ulcers. It can be normal ulcer, it can be a bleeding ulcer. You can see that this is normal structure. Can you see this structure? Now this has been destroyed, this mucosal barrier has been destroyed and the ulcer has been formed. If you have to treat that patient, and for that treating that patient you require that reason why that acid peptic, that mucosal barrier has been destroyed, why acid peptic digestion has taken place. Either it has been produced excess HCl or it has destroyed the mucosal layer. So we have to stop production of HCl or we have to reduce in somehow or other, and for that biochemistry knowledge is required. Pharmacology mechanism of action. How does this omeprazole, what you take, or ranitidine, how does it work? See again endoscopic view of the gastric ulcer. The more you see, the more you will see. See, can you see the how many days he has been, how many months he has been suffering from? So parietal cell basically it produces HCl. HCl is produced. Here you see that normal that carbonic anhydrase is from bicarbonate, and this hydrogen is entering into the lumen of the stomach to produce hydrogen ion, and bicarbonate is formed and it is thrown into the blood, and in return chloride ion enters into the lumen of the stomach, and that forms HCl, and that is again under the control of histamine, and this hydrogen ion is pushed out of the cell, stomach cell, into the lumen through a pump. That is, remember, write down hydrogen potassium ATPase pump, and that is what is proton pump. These gastric parietal cell are containing H2 receptor block, H2 receptor, histamine 2 receptor, that's why the ranitidine or famotidine, whatever we give, that blocks that histamine, this receptor is blocked. So we are partially inhibiting, because gastrin we are not encountering and acetylcholine we are not encountering. So these will act. Partially we are blocking this histamine action on the parietal cells. So some reduction will be there. Now if you use a proton pump inhibitor, which reviprazole or omeprazole is that, it will completely nearly stop the HCl. So is it desired? HCl is having some function. So therefore, don't jump to give proton pump inhibitor in the initial stage itself. Depending, you just inquire what is the degree of that patient. I mean, if it is a small one, then give an anti-H2 receptor blocker first. Reduce, observe for some time. That may be patient may be healed. Don't jump every time to proton inhibitor. I am telling you. The proton pump inhibitor basically stops, completely blocks. So as I told you, this, it basically produces HCl, and this proton pump inhibitor, the drugs are extensively used. Nobody uses histamine receptor blocker. Because earlier again this was discovered early in the early 80s when these drug proton pump inhibitors were not there. But most of the patient is going to get cured with that drug. So, mechanism of action of proton pump inhibitors you know, mechanism of antihistaminic drugs you know. How does it act? And I told you earlier also, one student was, sir, we are not understanding pharmacology is very difficult. Because if you know the biochemistry, then what you will do, basically that biochemical path has to be blocked by the drugs. So, this is schematic diagram of small intestine. So, again liver anatomy, gallbladder anatomy has been told. This is the problem, no? Then you just read it. If I require, I will take one SGD class after you read GI system anatomy. So, from the basically from two sides of the liver, two common hepatic ducts are formed. The common hepatic duct, it enters into the bile, and in the gallbladder, the bile is concentrated, and that concentrated bile is thrown through the common bile duct to an ampulla veter into the second part of the duodenum in combination with pancreatic duct. So, this is the difference between a non-medical person and a medical person. Non-medical person will never do anatomy, they will don't know the anatomical structure, and if you do not know the anatomy and if you try to attend biochemistry, situation will be just like a non-medical person, nothing more than that. Once you cover this thing, then it will be easier to understand. So, pH of the gallbladder is some different thing, and pH of the common bile duct is something different, because that must produce a conducive environment for alkaline enzymes medium to act. Again, you see the second part of the duodenum, a number of hormones are active just like stomach. So, that stimulates the formation of pancreatic juice. Note down this one. Pancreatic juice is strongly alkaline. It contains these 4 enzymes. One important point I want to highlight, carboxypeptidase is present in pancreatic juice, but amino peptide is present in intestinal juice. Both are exopeptidases, but one is present in the pancreatic, one is present in the intestinal juice. Remember this one. Achha, absorption, coming to the in one slide I have made it so that you understand. Occurs in the small intestine. It's an energy requiring process. Remember, transport across the membrane, it's an energy requiring process. It is a ATP sodium dependent or sodium independent. Sodium dependent in dependent form of 3 amino acids are absorbed across the intestinal mucosa. And there are 5 different transporters, carrier mediated. One is for this, you note down. One is carrier for neutral, one is for basic, one is for immuno, one is for acidic, and one is for beta alanine. And they compete with each other for absorption. Because at the end we have digested, we have broken down into amino acids, that amino acid needs to be absorbed. And these are done by energy requiring process. Mr. Cycle, gamma glutamyl, this comes as a short note. So, that is involved in the absorption of amino acids. So, it is absorbed for neutral amino acids. You will get this picture in the book. You try to remember that thing. Only thing I want to highlight, the transport of one amino acid across the membrane, it requires 3 molecules of ATP. So, it is a high energy requiring process. How 3 enzymes are required? I am telling you this process, what is happened in the transmembrane protein is there, that is glutabil gamma glutamyl peptidase, transferase or transpeptidase, that combines with amino acid form gamma glutamyl, and the cysteine glycerin molecule is released. Now, this gamma glutamyl cyclotransferase basically transfers the amino acid. So, amino acid which was present outside the lumen is now inside the lumen. From that it will be transferred to veins, that is finally carried by the portal vein. So, where from 3 ATPs are required, See, gamma glutamyl is basically glutathione. The glutathione has to be regenerated. It's a complicated process, and you see how 3 ATPs are remembered. At least if you cannot remember this thing, write down this one, Mr. cycle, about Mr. cycle. This at least 2 steps how the amino acid is transported from extracellular phase into the intestinal lumen. That's what. Only thing, that's why this is the minimum amount I have required for single amino acid. 3 molecules of ATP will be required. So dipeptides and tripeptides, they enter the brass border. Normally it is cleaved in the intestinal succus entericus juice into peptides. If by chance if dipeptides and tripeptides are enters into the lumen, into the intestinal cell, there is also dipeptides and tripeptides that will break down. So that will again finally in the portal system we will get amino acids. But you have seen some of the proteins foods we are having some allergy, because these proteins are not absorbed in just from amino acid in the form of amino acids. There are some transcellular or some paracellular, means within the between the cells there is some space. Through that space, some partially digested proteins or polypeptides, small fragments of polypeptides enter into the portal vein, and that causes food allergy. Normally amino acid do not cause any amount of immunogenicity, no antibody formation, no allergy. But partially digested, partially if it is formed in the partially digested form, so if that enters into the transcellular, paracellular, that causes a food allergy. So this is basically summary of the entire protein digestion. One thing I want to highlight, maybe this thali is very good to look at, and of course anybody will release, but normal energy required for the diet, carbohydrate 6 to 70%, fat 25 to 30%, protein 10 to 15%, and it constitutes 1 gram per kg body weight for any individual. Remember, 1 kg means this where somebody is having 60 grams, 60 kg, 60 grams. So if you take a 60 gram fish or 60 gram meat, that will not meet the requirement, because it is having 70% water. If you take fish as such, without bone, without fat, so 70% water is present in that food. So to get 60%, you have to take nearly 200 grams of something. Like milk, if you take chana, paneer, whatever you are taking, that contains some amount of water. So in terms of absolute content, do not get confused that I have 60 gram body weight, I have taken a 60 gram fish, means or 100 gram fish means it is, I have taken, no. Basically 70% of that fish is water. Now if you take, many of you are in the habit of taking excess amount of protein. If you take excess amount of protein, what will happen? Normally whatever food you take, you do not have a calorie deficiency. If you take excess protein, it will serve no purpose. It will serve no purpose, I repeat, because there is no storage form of protein. No storage form of protein. Like if you take excess carbohydrate, that carbohydrate will be stored as glycogen. If you take fat, that will be stored as in the forms of lipids. But if you take excess protein, more than what you require per day, that excess protein will be absorbed. It will be catabolized to urea, and you will excrete that protein through the urine. So what is the purpose of taking some extra amount of food? See, carbohydrate gives 4 calorie energy. Protein gives also 4 calorie energy. But the cost of protein is much higher than cost of carbohydrate. So you take the energy, get the energy from carbohydrate. Don't forget to take adequate amount of carbohydrate. Do not try to take protein from energy, because if you take too much of protein, you can ask that if I can take. Yes, you can take. Only thing is that if you take protein too much, then the stomach will, the entire protein will be digested and will pass a hard stool. We want normal activity of the intestine also. The intestine, we must form a normal stool also. So for that fibers are required. and if you take that, I will get the entire energy from protein, just like a carnivorous animal. You can definitely, but at the end you will have no storage of carbohydrate, no storage of fat. Is this part clear?